And we're back. Hi everyone, welcome back to Wildlife Cake and Cocktails. This is your friendly neighborhood wildlife nerd, Yanni Torcola. We're using the powers of satellite to come to you guys this week from interstate for our first audio show. Now, in honor of our guest who is working on an innovative method to hopefully protect northern quoll populations from cane toad, I'm drinking something called a hop toad. It's mostly rum, apricot, brandy, freshly squeezed lime, ice, and some bitters. And god damn, it tastes like chewing on a cane toad, and I think it might have the same effect after a few of them. I've also got a lovely bit of chocolate ganache cake here. Now, being a snake catcher, I'm obviously driving around a lot, and sometimes you've got to eat. If anybody's up in the Gap and feels like going to Bannerton Bakery, man, they got some nice pastry, they got some fantastic croissants, and I got this beautiful chocolate cake here from them, so can't recommend them highly enough. Enough about what I'm doing, though. Let's get straight into it. Our guest this afternoon is Mr. Chris Jolly, working on a PhD at the Ecology and Evolution Labs in the University of Melbourne School of Biosciences. So, Melbourne, that being said, he's coming on the line right now from the Northern Territory. Chris, how you doing, mate?
Yeah, I'm good, mate.
Been busy up there?
Oh, relatively so, yeah. Like, I had to delay stuff today because we've had a few snake calls, but, yeah. I mean, even in winter here, when you guys have got nothing to do, we've got 5 calls a day.
Oh, Jesus. I'm lucky if I get one. I'm seriously struggling.
But it's not good for us because we've got a government contract, so we get paid regardless of how many calls we get. So we prefer to have none.
Oh, wow. So it's completely the opposite from what we're doing down here, struggling in winter and having to pick up educational. That's kind of... I might have to move up that way someday, maybe when I get a little bit older or something. But not to horn in on anybody's territory. Right?
Yeah, no, it's all good, man.
How long you been up there in the NT now, mate?
This time round, I think about 7 or 8 months.
Righto.
I've moved here from Cairns, but I have lived and worked in the NT previously.
Okay, cool. So whereabouts are you at the moment? Where are you calling us from?
Oh, I'm calling you from the balcony of my house in Stewart Park, which is maybe 2 kilometres from the CBD.
Oh, right near Darwin there.
Right near Darwin, but you wouldn't think so because we've got greenbacked Gerigines calling in the background right now, and slaty grey snake calls across the road.
Man, it just gets so green so quickly, so close to the city up there. It's absolutely phenomenal.
Oh, it's lovely. The Northern Territories, especially the top end around Darwin, if they didn't burn it, it's rainforest.
Yeah.
But because they keep on burning the outer suburbs, it's savanna. You stop burning it, turns to rainforest, but rainforest is almost excluded across most of the top end by fires.
Yeah, right. Otherwise it would creep in pretty quickly, hey?
Oh, yeah, that rainforest is lovely.
Yeah, awesome. Speaking of slaty greys, did you see the Territory Wildlife Park's nice albino that they posted, I think, today?
Yeah, I was on call that night, and the snake was... The picture of the snake was posted on a Facebook group up here, and everyone was tagging our snake catching. We do the after hours snake catching business, Darwin Snake Catchers,
right?
And everyone was tagging Darwin Snake Catchers in it, saying, "Call them, they'll come and pick it up. They'd be very interested in that snake." Blah blah blah. And and then I got a very unusual snake call where a woman was talking to someone in the background. So I picked up the phone and said, how you going, Darwin Snake Catchers? And she was talking to someone else, and she said, oh, it's just over there. It's just over there. It's just moved. Sorry about this. I'm just speaking to someone else. Give me 1 second. Oh, the snake's just crossing the path over there. And she said, sorry to bother you, mate. Another snake catcher has turned up.
It's always the way, isn't it?
I'll let you go. And I was like, yeah, righto, thanks, because I was in bed watching a movie. And I was like, well, that suits me. But I know there's no other snake catchers in Darwin, right? Like, we're the only people with the contract to do it.
Right.
So I was like, well, it means I don't need to drive out to bloody whoop.
Yeah.
But yeah, it looks like it may have been that snake. So
you may have nearly gone out and been the one to bag up that lovely albino slaty grey.
Yeah. So and also in the NT, the way our contracts run is we've got a permit to keep any snake we want if we want to. We never do. We're not interested in it, but we can keep and sell any snake we catch, which is pretty ridiculous.
Yeah, it is. But God, I can imagine some people would have wanted
that snake would have been ours if we went to that snake call. But I reckon I probably would have just released in the wild just to piss off those captive keepers.
Oh, that's harsh, man. That's harsh. You're not going to maybe pop out a couple of little hybrids there to keep in the line, no?
Oh, it's not my call. Territory Wildlife Park have got it, but I reckon we might suggest to them they should attempt to breed it. I don't think they've got any other slaty greys at the moment, but
yeah, right.
I reckon they should give it a crack.
Phenomenal looking snake, beautiful albino.
Oh yeah, lovely.
Do you get a lot of slaties in the area?
Fuck. Yeah.
Absolutely everywhere, all over the place.
It's absurd. The number of slaty grey snakes in Darwin is ridiculous. Every night.
Quoll Dam is just covered in them. There's more slaty greys than there is around.
Oh, for sure. Like Greg Brown that's out there, I think he marked 10,000 slaty greys before he ever got a recapture. He thought they were an annual species that just bloom and fade away. But it turns out it's just but like that's Quoll Dam, right? Like that's in the wild. But I'm talking about suburban Darwin. Like I'm like I can see the city. I'm surrounded by houses. These are small city blocks. Yeah. And on this street alone, I've probably gotten 5 slaty greys in the last month.
Wow.
Like every night, someone's got a slaty grey in their backyard, and we have to tell them, right? Like I know I'm just diverging from the topic of quolls, but quolls are boring. Snakes are much cooler. We have to tell people, right? Like it's very normal to have a snake in your backyard. Would you call us if you saw a peewee or a green tree frog? Like we're trying to change people's attitude because in Darwin, like unlike Brisbane and the East Coast where like some of the snakes are highly venomous, we virtually never get a venomous snake. This place is just littered with pythons and colubrids.
Yeah. Well, look, I mean, that's kind of the case even down here. The majority of our call-outs, the majority of our call-ins are definitely for carpet pythons and tree snakes. Now, that doesn't mean, like, we're going to go out for most of them. A lot of the time, as soon as it's identified and people kind of, you kind of address the fact that, probably about, at least 1 in a dozen houses has a python somewhere on it. And they're really common, and they're doing a great job keeping the rodents down and keeping the spirochetes out of people's brains from the rodent urine. So, I mean, people come around to it pretty quickly. So.
For sure.
Yeah. Hey, look, we better start talking about what we're here to talk about, some quolls and stuff. But just briefly, mate, I'm always interested. What got you into ecology and wildlife conservation? I know you're a bit of a reptile fanatic, kind of like myself. Is that kind of your what was your kind of first interest?
There's just something really wrong with me, like a lot of us.
You just can't stop chasing reptiles around.
Well. I suppose that's it, but we love to have a fucking miserable life. It's just bloody hard work.
It is.
I just have always liked reptiles. I don't know what it is. Like, I've always liked animals. I've found them so bizarre.
Yeah.
Like, they're such weird, different things, aren't they? Like, so different to us. But when I was just a little child, I was obsessed with sharks. I've always just been one of these really obsessive people, like, I get a new interest and I can't stop until I've learned everything about it.
Latch onto it a bit compulsively and just start digging into all the cool stuff that we know about it so far. And
that's it, yeah. Sharks and then dinosaurs, and then when I was about, 5 or 6, a kid brought a pet snake to school, and I grew up in inner city Sydney, so I'd never encountered a snake before.
Right.
As soon as I discovered you could keep these things as pets, my world changed.
Yeah.
and I never grew out of it, unfortunately.
Yeah, well, look, I'm fairly similar in that regard. You know, I moved over here from Europe when I was 2, and upon seeing reptiles walking around the backyard, that was it. It was game over. I needed to know everything about them, and haven't really stopped since. And so you've mostly done a lot of. I actually know you from some of the reptile pages online originally.
That's true. Yeah.
And I think that's how we got into contact about some of the research that both of us were working on different angles.
Yeah.
And I think so. You're now working heavily with Quolls.
Yeah, I am. You know, like, people care about mammals. They find that sort of stuff. So if you want a job, you go where the money is, don't you?
Well, that's true. And they are a very enigmatic, fluffy, cute species that's going to get a lot of attention. But I do like the fact that you guys are using the opportunity really, really to test some of these adaptive evolutionary principles that we're going to get into.
Yeah, I mean, I'm only being silly. The reality is, I don't actually. I love reptiles, right?
Absolutely.
But I don't actually mind. I like reptiles because they're cool.
Yeah.
But I'm interested in good science, so I don't actually care what species I work with if you can do good science with it.
That's right.
Some animals are easy to do that good science with than others. So the opportunity was given to me. I got approached to do a PhD with Ben Phillips, who's like my supervisor, and if he ever hears this, he'd be embarrassed, but like, he was my academic crush for fucking years. So when he asked if I wanted to do a PhD on Koalas, I was like, sounds all right, but yeah, I'll do a PhD with you. But the science is just really good, so that's why I work on Quolls.
Well, that's right. Well, look, I mean, I can also kind of relate. I did a year and a half of honours on sea squirts.
Oh, lovely.
Yeah, on an invasive ascidian that looks kind of like a malformed brain with 2 holes in it. That's also invasive and just encrusting and infesting the native ascidian. So it's
true.
Yeah, not the most pleasant organism, Styella plicata, but I got to work with a really, really great lab there with Cynthia Reginos in the functional and ecological evolutionary lab at UQ. So I had the opportunity was there, and it was a great opportunity. It would have been a fool to pass it off. So,
invasive species are interesting. They like open up a world of science that a lot of native species don't. Just like these interactions, like that they present, and functional ecology and stuff is really cool. I mean, it's often depressing because there's not much we can do about it a lot of the time.
It is, but it's sort of a unique evolutionary challenge as well that a lot of them haven't faced before. So it is fascinating to see how native populations deal with it, and what we can do to help them deal with it anyway. So, all right, cool. Well, look, let's talk about northern quolls a little bit. So, just a little rundown for the audience or whoever's listening, and correct me here if I'm wrong at any stage, Chris.
Sure.
Dasyurus. So they're the dasyurids. And that includes 6 species, including all of the quolls. They're marsupial predators. They've got a pretty wide variety of prey. They'll take some fleshy fruits, a larger variety of invertebrates and vertebrates. So they'll eat a lot of frogs and lizards and figs and flowers and things like that as well occasionally. Arboreal and terrestrial, fantastic climbers. 8 teats apparently. Can have 4 to 8 young. Sometimes they have more, but apparently the young have to kind of fight it out for space, and not all of them make it. According to the International Union of Conservation of Nature's Red List, they are endangered at the moment. They were already struggling from feral predators, cats, dogs, foxes, and the like. Fire in the area. If not managed correctly, severe fire, of course, making things worse by destroying their cover. And since then, the cane toad invasion has slowly been moving across northern Australia and entering the areas where northern quolls live. So 2006 was actually the Territory Wildlife Park, I believe, that established a breeding program with then work by Rick Shine and a bunch of others in training toads to be aversive to cane toad flavoured baits, and then hopefully cane toads in the wild, with release of Toad Smart Quolls in 2010, which is an ongoing project at the moment. How'd I go? Did I miss anything important?
No, pretty good. Yeah, you got pretty much everything right, eh? You seem to know more than I do about quolls.
Well, I've got some help on the background research here. I'm just reading from a list.
No, fair enough. The only thing that I would say is, other than the fact that I don't think it's foxes. There's no foxes in North Queensland that are likely to have impacted. And also, you said dogs. And I'm not sure
Dogs/dingoes?
Dogs. People are afraid to say that dingoes can have an impact on an already declining population. And that's what we found with our most recent release, right? So the start of my PhD, the initial intention was for me to try to answer this question that's been bandied around for a long time. So it's been in the media. I mean, even David Attenborough has spoke about it, that we can train northern quolls not to eat cane toads. We can release them into the wild and they will train their young, and it will influence long-term recovery. We do not know that. There is no evidence to suggest that. And that's what I wanted to get to the bottom of. We know very, very confidently that we can train quolls not to eat cane toads. We release them, they go into the wild, and those individuals do not eat cane toads. So
great short-term success.
Oh, yeah. Well, yes.
Whether or not that passes the test of time or down generations.
Well, yeah. So there was one of my supervisors released before me. It was a previous PhD student. Released quolls into northern Kakadu. They were all trained to avoid toads. The vast majority of them survived, and they reproduced in the wild, and their young survived as well. But and when they did a parent analysis, I think it was found that those females actually reproduced with wild existing quolls. So there's no way to know whether they actually train their young to avoid toads, or whether
there was adaptive divergence already in the population.
Yes, and presumably if there's a quoll that's living freely in the wild, it doesn't eat toads. If it did, it'd be dead. So
sure, as well, especially if the invasion funds passed through there some time ago.
Oh, indeed. So I went to address that. We released 30-odd quolls in southern Kakadu, where quolls have gone completely extinct. So we knew that there was going to be no incursion of wild genes into the population. We had controls, so quolls that weren't trained to avoid toads, and we released them, and all but one of the controls ate a toad on the first or second night and died. If you don't train them, they die almost immediately. But what we didn't anticipate is that basically all of our quolls that were trained avoided toads but got eaten by dingoes.
Oh, bugger.
But what I believe happened was that the conservation of quolls in the Northern Territory was to collect up quolls ahead of the invasion front, and they were put on some remote islands in Arnhem Land as a satellite conservation population, and they've done exceedingly well on these islands. Like, they went from 45 individuals to over 3,000. This is an estimate anyway. Over 3,000 in 4 or 5 years. So they exploded. They're doing really well. The next step is you release them on the mainland, right? But this is a predator-free island. So they've gone 13 generations now without any interaction with cats or dingoes. So that was the next question to answer for me. Have they lost their aversion to dingoes? And so I ran a bunch of experiments at the Territory Wildlife Park, and sure enough, compared to mainland quolls, they don't recognise dingoes or cats, whereas their mainland counterparts do.
Wow. So while training them to be, I guess, toad averse,
yep,
they've lost their dingo or cat aversion, their predator aversion, just in their time on the island.
Yeah. Well, I mean, quolls, it's great that they have annual generations, because it means you can they really rapidly evolve.
Yeah, sort of what's a 1 to 2 year lifespan for males, 6 months to weaning, maybe 3 year lifespan for females, and again 8 teats, so up to 8 young, with 6 months to wean. So that is a very rapid generation rate.
It's essentially the males breed once in their lifetime and die. The females typically breed once and die, and some, I think about a quarter of the population may get a second.
Maybe get lucky, yeah.
Yeah. So you can have this really rapid evolution, but the flip side of that is we took quolls from the mainland where it's a predator-rich place where shy individuals are selected for because they avoid predators, and we put them on a resource-poor island with no predators. So the selection regime is flipped on its head. So now
they need to get out and eat everything that they can see and not worry about predators so much.
So the bold individuals prosper. Not only do they lose their aversion to a predator, which is costly when it doesn't exist, you know. If you're hiding for something that's not there, you're losing out on resources. But
absolutely.
On top of that, they're running out of food, so they have to. These quolls on the little islands have to forage on the reefs and stuff to get enough food. So it's like these really bold quolls that are favoured, and then we cherry pick that even further by going and putting out traps, and only the ones that are super bold that are willing to go in the traps.
And select themselves.
Yeah, I'm just speculating here, but it seems to make sense anyway.
Yeah, no, of course. Wow, so you've actually found them foraging on the reef, like on little reef flats or atolls?
Yeah, well, I haven't, but there's these quolls that came back to the Territory Wildlife Park.
The island quolls, obviously.
Yeah, the island quolls have worn their teeth to nothing from eating hermit crabs and I don't know what else, corals and...
Wow.
Yeah.
What little savages. I love them. They're fantastic little beasts.
They'll eat absolutely anything, including toads. And they're great. They're great little animals. They are ferocious, though.
How are your hands looking these days?
Oh, I stay away from them. I don't know. I avoid them at all costs.
That's pretty smart. Stick to handling snakes, hey?
Yeah. Oh, venomous snakes are easy compared to mammals. Mammals are terrifying.
Yeah, well, the way that they can use their feet and claws to grab you and then sink those teeth in, man, it's just a whole different game. All right. Well, look, I guess we should move on to the next bit of your PhD. So the thing that I find really interesting is, look, with a lot of conservation genetics, you know, you're really working hard on molecular stuff. You know, you're looking at gene flow or sequence diversity or haplotype diversity to see if there's been reductions in population or connectivity or population bottlenecks. You're looking at population genetic structure and things like this. A lot of it's quite it is hugely expensive and time consuming, and then a lot of the time you may not even get what you want out of it because you may not have the markers that give you enough coverage, or there may not be variation, or there may be unprecedented variation that you didn't expect. It's always very interesting, but I kind of like just reading a little bit about your work on this PhD. It's interesting how you guys are kind of using the theory of adaptive evolution in an applied way without having to sequence every animal in your collection. I mean, you totally could. I'm not sure if you are.
Oh, it's getting done, but we're ecologists, right? So
that's right, yeah.
So we don't know anything about that. So, and nor am I that interested, really. Like, at the end of the day, if it works, it works. Like,
yeah, if it works for conservation purposes, then it works. So look, let's start talking about this. Let's talk first a little bit about assisted gene flow and targeted gene flow. So as I understand it, assisted gene flow was kind of the idea of helping populations adapt to things like climate change. You may have, for example, a population of eucalyptus trees, or more recently, there's work being done on some small skink species that may have thermal tolerances that are going to be, those populations are going to be at risk in future climate regimes. There may be other populations that are already pre-adapted to those hotter conditions that are, separate subpopulations perhaps of some of the less adapted populations, and you can perhaps shuffle a couple of those individuals over, or some of the genes from those populations over, and help them adapt to future climate change. So that's assisted gene flow. Very, very interesting. Now, targeted gene flow, what you guys are doing there with the Quolls takes it a little bit further. So the idea of targeted gene flow is basically to use this assisted gene flow technique for other things, such as maybe new pathogens or parasites, or in our case, invasive species, not only to either slow them down, but to maybe help native species deal with a new invader in their population, in their range. Sorry. So, is that about right?
Yeah, I think so. Yeah, it's targeted gene flow is just a bit more aggressive than assisted gene flow.
Right. So you're really looking for populations of northern quolls that have had many generations of cane toad around them, and their immune systems, their digestive systems, their behavior, all of the genes which are involved in those are being selected to be, I guess, toad smart.
So we have no idea what has been selected for. All we know from the other side. I can't talk completely about this topic. So there's another PhD student that's doing more of the more directed stuff with targeted gene flow. But what she's found is that previously it was looked to see whether quolls had resistance to cane toad toxin. They don't. So quolls that have lived in symmetry with toads for 80 years in North Queensland have no more resistance to cane toads than ones from Western Australia that have never seen them before, and that resistance.
So biochemically, as in terms of the toxin.
Yeah, to the chemical, to the poison, they have absolutely no tolerance to it. They die without even eating it. They just bite it and they die. But when Ella compared quolls from Queensland with toad-naive quolls from the islands, they show that they just don't eat them, and their young don't eat them either. So there's
for whatever reason.
For whatever reason, they just don't eat them, and whether that's as specific as they don't like toads, or they are just more wary, and so they may get a small amount of a distasteful encounter with a toad. We don't know that, but all we do know is that they survive in a toad-infested landscape. And I've seen in North Queensland, while I was working there as an ecologist, we put out camera traps, and we'd have a cane toad set off the camera trap, and a northern quoll would run up to it, excited by the movement, and as soon as it came within a few centimetres, it would recoil and know that it shouldn't touch that, whatever it is, and then the toad would hop again and it would get excited, but it wouldn't actually make that fateful leap into actually biting it. So we know that they can survive we know that they don't eat toads, and if they did, they'd be dead, and that's why this has presumably happened, because all the ones that will eat toads will die, and the ones that don't survive and pass on, whatever it is, to the next generations, and then so on. So what we've done with this assisted gene flow project is crossbred at the Territory Wildlife Park, Queensland, so toad-averse quolls with NT quolls, and we've released these quolls on an offshore island, and I did that just a month or so ago, and we've released pure NT quolls, NT Queensland hybrids, and some pure NT males. So what people might say is, why wouldn't you just release the Queensland animals ahead of the cane toad front so they avoid, so they don't eat toads from the get-go? Well, the assumption is there's some local adaptation to a broad range of other things in the environment other than toads, which may be important. So these animals are from the NT. There's a lot of other environmental factors that they may pre-adapted to, and all we want to select into them is their aversion to toads. So it's been modelled by Ella and Ben and how it would work, and now we're actually seeing if it works in the field, because that's the important thing with conservation, right? Like, it's all good.
Yeah, actually getting it out there and getting it into action and saving some animals, right?
Yeah. Wonderful.
So, that's really interesting. The hybridization issue is is quite interesting to me. So you're basically hoping to get some of the positive adaptive traits of the local Northern Territory quoll population and some of the toad smart Queensland genes, I guess. And get some, I guess, what you might call hybrid vigor.
Yeah. Well, I don't know. We'll see. We don't know what we're going to get. But so this first generation that we released into the wild, we needed the we needed the NT animals to survive as well. So everyone got trained using the same technique for any quoll training, so that they survived and reproduced. And what we're interested in is the next generation that's born in the wild. But when we trained them, when Ella trained them, I was in the field at the time actually doing a pre-release survey. It looked as though, so the NT animals will happily eat a cane toad, and that's how we train them. We give them a small cane toad with a nausea-inducing drug, and that prevents them from eating a fatal toad in the future. So all of the NT ones were willing to eat that dosed cane toad, and of the hybrid ones, there was about 50% that were willing to eat those dosed cane toads and get trained, and then the pure Queensland ones, I think only about 8 to 10% were even willing to
eat one in the first place.
So we couldn't even train them because they already knew not to eat them. So these are animals.
So there's obviously some serious selection acting on that population.
Oh, for sure. And this is like second generation captive born. So there's no selection going on in the last couple of generations. It's just that it's persistent.
There's definitely no learning, no social learning from other quolls to avoid them. It's innate. It's something heritable by the sound of it.
And that's what I've always suspected. Like, I think it's a fancy tale. The whole teaching their young. They're not smart animals. They're stupid. They're like a reptile. Like, what are they telling each other?
Look, marsupials, while they are fantastic animals, they are proto-eutherian mammals. They are a little bit older, generally a little bit smaller brained, you might say.
Tiny little brains.
Yeah.
But they're beautiful creatures.
Absolutely.
They're just really dumb. And that's fine. I know some beautiful people that are really dumb. Yeah, but they can still evolve.
I've got some friends who are very, very similar to quolls.
That's great.
Well, dude, that all sounds really interesting. So, you guys are releasing them out there on Indian Island, which no quolls there before you guys arrive.
Well, not in the recent.
They're all wiped out by toads. Is that correct?
Well, we don't even know if they were ever there. No, truly, it's an introduction. It's not a reintroduction. It's an invasion.
Okay, so it is a truly experimental island population.
Oh, it's an invasion, and that's actually what my PhD is about. This targeted gene flow is Ben and Ella's little baby, and I'm doing some work with it, and I will be a co-contributor to the papers that come out of it, but it's part of Ella's PhD. What my PhD is about is more about predators, and what my PhD is what is the cost to the endangered species and the host environment of conserving endangered predators on islands, because what we're essentially doing is analogous to releasing a feral cat population on an island.
On an island that may never have had quolls on it in the first place and may have endemic species there, which...
let me make this clear. It doesn't have endemic species. It's only 500 metres from the mainland, and we've done rigorous surveys to ensure and looked at genetic samples of cryptic species. So it doesn't have endemic species, but
in principle, if somebody were to make that error,
lots of people. It was done in the past with northern quolls on these islands, and those islands weren't surveyed for cryptic reptiles. And the better our genetic techniques become, the more endemic species we find, cryptic endemics on islands.
Absolutely,
yeah.
So
is the conservation of one species worth the cost of another? That's a real ethical dilemma. I think not. So it's a really unique opportunity that I have to look at what are the consequences to the environment of releasing northern quolls for conservation and for a neat experiment, but it still comes at a cost, right?
Yeah, well, that's a phenomenal point. What are the conservation costs of conservation? A lot of people don't consider that a lot of the time.
No, they go to one-track mine and they're like, we got to save this species. Like, the reality is, animals come and go. Like, northern quolls in North Queensland are rebounding from cane toads, and that's how we can run this targeted gene flow thing. So if people were to start putting quolls on islands all around coastal Australia, like, there will be species that go extinct because of that.
And they are phenomenal predators, as we mentioned. They'll take birds, they'll take anything that moves.
So it'll be interesting, and we'll see. What I'm really interested in is the evolutionary the evolutionary impacts of quolls on this island. So this island is absolutely ridiculous in the abundance of mammals compared to the mainland. Like, we've done preliminary assessments, and there's about 60 melomys per hectare there. So, every 100 by 100 square metre plot of land, so, literally the size of my backyard, there's 60 melomys on that land, in that area.
Melomys, for the listeners, by the way, are a small
rat.
Small native rat,
Beautiful little fellas.
Gorgeous.
But totally naive to mammalian predators. They've never seen a cat or a dog or anything.
Right, so they'll run from reptiles, but...
there's not even many, there's not many predatory reptiles on the island either because of cane toads, basically. So these melomys, you can just walk up to them and pick them up in your hands. Like, they are so bold and so naive to the dangers of a predator.
Well, that's island populations, right? The same thing that happened with the quolls happens naturally.
It does. But what happens if a seed dispersing rodent goes from these massive abundances to nothing? What are the really...
You've lost a potential vector, you've lost a potential prey item for something else local that you may not have seen yet. Well, that's really interesting, because I was reading about some of the risks about targeted gene flow, such as, translocation having a huge failure rate for those non-local adaptions, potentially vectoring parasites or diseases, or outbreeding depression, which is kind of the opposite of hybrid vigor, where instead of getting the best parts of both sides of the hybrid, you kind of break up some important gene complexes which are kind of working together. My impression was that is more likely than hybrid vigor, right? That outbreeding depression is more likely than hybrid vigor.
Is that right?
I'm not sure. I guess we'll have to look into that.
Like hybrid zones of two closely related species would suggest that hybrid vigor is the less likely occurring thing.
Right. Well, especially if it's forming a barrier, then absolutely, yeah.
Species maintain themselves through the hybridizations being maladaptive.
Well, I mean, hybrid zones can be temporal as well, and they can be diffusive at the time while you're seeing them. But I mean, that's another very good point. But look, I didn't kind of think about that idea of another risk of being, the local ecosystems around.
Yeah. And I mean, a lot of people, that's their major criticism of the project. And I think it's important to address that. Like, the quolls are being released. It's an interesting question to run, but at least we can say, well, we know what the consequences are because there's more and more animals getting released on islands. And the other thing is there's a lot of animals being conserved on islands. And if you're, not just islands, predator exclosures and in captivity. And if you're conserving these animals in this way and you're removing them from a threat that they're no longer under the selection pressure of, just like the quolls losing their aversion to dingoes, an animal that it evolved with. It's been evolving with for 3,000 years, and then 13 generations on an island, and they have absolutely no way of dealing with them. Like, is this really a good way of conserving? Like, to put animals under a completely useless selection regime that makes them they have absolutely no use for reintroductions to the mainland now because they've been on an island for
so long, for so many generations without the same selective pressures. Well, I mean, it is interesting to see whether or not that hybridization procedure could even maybe assist in those reintroductions because you may not be able to release those island populations back unless you hybridize some local adaptive genes from wherever you're going to put them.
Or you just stick a couple of predators on the island.
Well, that too, definitely. But bring back those selective pressures and see what happens.
Arid Recovery is actually doing it at the moment. It's really cool. They've introduced D6 cats to some of their predator exclosures.
Right.
Well, I don't know if I should be talking about this. No, it should be fine. And like bilbies are seeing their mates getting mauled by cats, and that's like improving their ability to avoid
survival
cats.
rates in the future. Yeah, absolutely. Wow, that's fantastic. Well, I mean, it's not fantastic that they're sending cats and they're shredding bilbies to shreds in front of their mates, but I mean, whatever works in the end. Yeah, that's right.
It's a violent world in
nature. That's it. Right? Yeah. Red in tooth and claw, I think is the old saying, something like that. How's it all progressing up there on Indian Island? You were up there, was it a couple of weeks ago, dropping off a bunch?
Oh, it's bloody lovely. And it's surprisingly close. Like, I thought I was absolutely I was radio tracking one of these quolls the other day, and I felt for 2 hours into the woodland in the middle of this massive island, and I was looking for my track back to camp, which was only about 3 kilometres away. It wasn't that far of a walk. And I just thought, out of interest, I'll see how far I am from home. And I scrolled the cursor across to where my house was, and I was only 42 kilometres away from home. Like, the island is not far at all. It's just in the harbour over from Darwin Harbour. But it's like, it's still remote. You don't see any boats go past when you're camping there. There's crocodiles on the beach. There's saw sharks, sawfish foraging in the shallows. You catch barramundi from where your tent's set up. Like, it's fantastic. Honestly, it's great.
Living it up, man. Well, look, I mean, it might only be 42 kilometres from home for you, but compared to a lot of people, home for you right now is pretty exotic as well, man. You're doing pretty well for yourself, living it up there, man. And the quolls are doing well. I saw some pretty fat-looking quolls on that island.
Oh, Jesus, they were a beast. I had no idea. When they came out of that box, those males, like the females looked a little bit more slender, but the tails on those males was just absurd. I was a little embarrassed putting those photos online.
They have been feeding very well, haven't they?
They better do their job and root those females. They might be too fat.
Well, let's hope they do, man. Chris, thanks so much. I can't wait to hear about how all this goes, mate. I hope those fat quolls do actually get some.
Yeah.
And I hope that you can come back and tell us about how the next generation goes.
Too easy. I'd love to. Yeah. No worries, mate. Appreciate your time, and thanks for having me.
Yeah, dude. Thank you so much for getting in touch with us. Always happy to talk to you. Guys, that's been Chris Jolly. Man, what a great podcast. I'm going to crush a few more of these toads, do my part for conservation. Thank you, everyone. We'll catch you next time. Cheers for joining us, and we'll see you soon. Bye.